Hello students, in this lecture video, let us discuss about different processes in molecular cloning or recombinant DNA technology. The first step is isolation of genetic material, that is DNA. So the cell will be broken down, means the cell wall or cell membrane will be degraded to release DNA. So we need DNA in pure form. So what we'll do? The first step is treating it with lysozyme if it is a bacterial cell to degrade cell wall and DNA will release. If it is plant cell, cellulase enzyme is used as the cell wall is made up of cellulose. And if it is a fungal cell and we want to isolate the fungal DNA, as the cell wall is made up of chitin, so chitinase enzyme is used. We also don't require rest of the molecules like RNA and proteins, so they will be also removed by ribonuclease and proteases. Finally, the purified DNA can be extracted through chilled ethanol treatment. Second step is cutting of DNA at specific location. By now we know that it will be cut with the same restriction enzyme as we are cutting our cloning vector with. Suppose we are cutting the DNA with EcoR1 enzyme. So the plasmid or the cloning vector will also be cut with the same to release the sticky ends and helping in ligation. Next step is amplification of gene of interest using PCR. PCR is the polymerase chain reaction which actually stimulates the process of production of billions of gene of interest. And because we want our recombinant DNA to be very much in high ratio to be obtained, so this process is carried out. Firstly, the DNA is denatured under high temperature. Then annealing of primers, that is DNTPs. And finally, extension, like the DNA polymerase does during replication. Similarly, under high temperature in the PCR machine, Taq polymerase will perform this step. So the end result is amplified gene of interest. Later on, this will be ligated with cloning vector. Next step is insertion of our DNA or our recombinant DNA into host cell. In the last lecture video, we have seen different transformation methods wherein you can insert the cloning vector along with the DNA molecule of interest into the host cell. After successful transformation, the next step is obtaining the foreign gene product. Main thing is expression of this DNA should be there and recombinant protein we need in large amount for commercial purposes. So generally, a continuous culture system is maintained and is used wherein the fresh medium, nutrient medium for the growth of the bacterial cell is added and the used medium is drained out so that the bacteria are always in exponential phase, means always they are multiplying. So large biomass we can obtain and further we can also obtain large amount of recombinant protein. So at commercial scale, there are large vessels in which the raw materials which we provide, that is nutrients, are biologically converted into specific products. It can be different organic acids, antibiotics, enzymes. So those vessels are referred to as bioreactors. Now generally, stirring type of bioreactors are used. Stirrer basically mixes nutrients. Oxygen parameters can also be regulated. Then pH, foaming, all these parameters can be regulated because there are certain ports which will help in providing samples as well as helping in regulating these parameters. Now, firstly, in the NCERT, I would like you to see the different two types of the bioreactors we are talking about. So, one is the simple stirred tank bioreactor where this agitation is happening. And in the second, the sparge stirred tank bioreactor through which air bubbles will be produced. So, all of this is to increase the surface area for the oxygen transfer. Lastly, the last step is downstream processing. Downstream processing means we need the purified recombinant protein. So we will separate and purify the protein from this bioreactor. So suitable preservatives will be used and obviously clinical trials have to be undertaken so that it is safe for human consumption.